edited 17 books, including all the viruses and the phytoplasma. And at present, he is working on characterization, epidemiology, and management of serial viruses and of phytoplasma. He has been awarded various prestigious national and international awards, like Overseas Broadcast Association Award 1996 from DST, President Award, Society for General Microbiology UK 1998, Best UP Agriculture Scientist Award from Uttar Pradesh government, and the Vigyan Ratna Award by CST UP government 2003-2004, and Jin Yu Q Award from China 2006, and IAP SIT Excellent Award from Egypt 2008, and Dr. Ram Badan Singh Vishisht Krishi Vaigyanik Puruskar 2014 from Upkar, Lucknow. And he has been actually, beside his research experience, he is deeply involved in editing very journals of national and international journals like Sugar Tech International Journal, and Phytopathogenic Molecules and International Journal of Phytoplasma. And he is also working as a Secretary General of Indian Virological Society for the last 1 decade, almost 1 decade. And he is a member of many prestigious scientific bodies, and he has visited more than 30 countries at various capacities. And recently he has edited 2 books, A Century of Plant Virology in India and Phytoplasma, Plant Pathogenic Bacteria. You will find that cover page has been displayed in our notice board also. So he has actually very edited so many books you will find in his credit. So I request Dr. Rao to please speak about your today's topic.
Thank you, Dr. Pan, for a nice introduction. Dear friends, students, as you all know, since morning you have been involved in the characterization of particular group of bacteria, very unique in feature, earlier known as mycoplasma-like organism. But in 1994, the International Committee of Mycoplasma, they have agreed because of a certain very unique character, this phytoplasma name was given in 1994. And since then, all those mycoplasma-like bodies infecting different diseases on plants now known as phytoplasmas. So as a phytoplasma, you can see now we have history of more than 30 years now, and in between every day now we have been formed all over the world, because in early days, because of lot of confusion, not very well awareness about this particular group of pathogens, very little progress has been made earlier. But after the discovery of PCR in 1990s, a lot of institutions where the facilities were available, they started working, and then we came to know the existence and classification and diversification of phytoplasma. And to your surprise, today we have more than thousands of plants known to be infected with different groups of phytoplasma. And on the basis, 2 different bases has been used for classification of this unique bacterial kingdom. That is one, as you know, as for all bacterial species, 16SR is the most conserved region. So that is the basis of classification of different groups. And another is RFLP, restriction fragment length polymorphism, that has been utilized for this finer classification of this bacterial kingdom. So we have been following, and still it is not very clear. So still every day with the progress of genome sequences, now we have more than 6 full genome sequences available. So we have knowledge about all the different genes associated with this particular bacterial genome. And now we have been utilizing more and more properties, host metabolic interaction, more gene function to characterize these the bacterial kingdom in a proper way as per ICTB for bacteria, fungi, and other plant pathogens, because I have prepared this because most of you must be having a little better knowledge about phytoplasma, but I have prepared this lecture only for the beginners, at least those don't have any idea, so that they could have understand much better, about this particular bacteria. As I told you, this is cell wall less parasitic bacteria, obligate parasite, very, very small in size with very small genome, and it is having a very particular shape. Any fungi and bacteria have a definite shape, of conidia or mycelium you can identify with, but this particular, it is known as pleomorphic, different, is not a regular shape, it keep on changing. That's why given the name pleomorphic, elongated, circular, subcellular, and it is located in the phloem cell tissues of the sheath tissues of the phloem cell, of the very, very specification, because it has its own, they don't have their own metabolic machinery, so synthesize their proper energy for their different metabolic activities. So they have to have depend on the phloem tissues in the plant cell, and hemolymph of the, and this is again very unique in the sense they can complete their life cycle independently in plant as well as insects. That is very, very again very, very unique. And along with, you can see its genome is, as I told you, very, very small, 600 to 1600 kb, and with very less GC ratio, less than 25%. And it is again only sensitive to only tetracycline group of antibiotics. So these are some characters that make it distinct from major main bacterial kingdom, Now come to little, I would like to give you a little historical background. You can see we have more than 100 years of history of phytoplasma existence in India. Starting from, you can see 1873 to 1955. But at that time, since we don't have knowledge about this particular group, so we most confused with viruses, or simply on the basis of symptom, we designated the disease and identify at that time. So these are some very, very important one. Root wilt disease of coconut. Still, it is these are, I have listed 4 or 5 diseases, which is for the last 100 years it has become problem, and again it is emerging regularly in the specific areas of their particular geographical distribution. For example, root wilt disease, you can see reported long back in 1874, and since then it has been very, very important and dominant in all south states, especially Tamil Nadu, Karnataka, Andhra, and is a very, very serious problem. We don't have any remedy to control this disease. We don't have any resistant varieties of coconut against this particular disease. And so far, very, very limited studies on epidemiology of the disease. That is again very important. Since we can't culture it, we don't have much knowledge about its biological properties, So we have to have depend now with only development of molecular tools. Now we have little progress, at least up to classification and taxonomy of these, so that we can plan further about management and other epidemiological parameters. Like sandal spike, again very, very important disease. Again, first reported in 1899, and since then, you won't believe, entire one of my colleagues recently visited, and he informed us in Karnataka all the natural forest, sandalwood forest has been ruined because of this phytoplasma spike. And we have only natural reservoir, only little left, saved in Mysore area. All the original natural reservoir of this sandalwood has been lost because of this phytoplasma disease. And earlier government, it is such an important economical value. Earlier government was never allowed farmers to grow this tree. But now seeing is importance of this disease, now farmers are allowed, now they are growing free nurseries. But this is the right time to work on this particular disease, to have more and more knowledge about other properties, how it spreads in nature, what are the other natural reservoir sources like insect and other alternative hosts, and what are the real losses caused by this phytoplasma. Then you can see yellow leaf disease of areca nut as equal as coconut, they are similar, originated near around, and again is a problem in all over southeastern states, South Indian states. Brinjal, little leaf. Since in you will happy note that first proof of electron microscopy of this disease was proved in this division by S.P. Rai Chaudhary and Professor Anupam Verma in 1940s, 1950s, 1950s and 1960s by Dean Stain and Dapis Stain. At that time, we don't have facilities for electron microscope, neither serology nor molecular biology. So only that time people used to confirm presence of phytoplasma by tetracycline remission treatment, either by this insect transmission or by major identification was done at that time by Dean's compound microscope and by using Dean's stain or Dapis stain. That is, one is fluorescent and one is very normal this microscopic stain. So this was again very, and still see first time reported in 1939-55, and still we don't have 100% resistant varieties. So you can realize the importance of this disease. Everywhere, any brinjal field if you enter, you will find a few plants infected with this disease. So it is very, very important. So now we are looking forward. Now we have well characterized on the all molecular basis, and now we are targeting full genome sequence to have more idea about different types of genes involved, and then we can target the virulence gene with help of various RNA silencing and other molecular approaches to control this particular melody. Sesame phyllodes again very, very important, very old, all over India it is distributed, causing again it is more dangerous than any brinjal. Sesame is more, plant is more susceptible, and again we don't have, some wild relatives are tried through crossing breeding program to cross the resistance, but we never got success. So it is again a major challenge before us. And then sugarcane grassy shoot. Again is is very important crop for Asian region, and especially Southeast Asia. We have this problem of phytoplasma causing sugarcane disease. Two phenotypic difference is there in symptom, but the phytoplasma is same. One is white leaf disease and one is grassy shoot. I will explain in later slides. Now come to how they are classified so far. What is little knowledge, updated knowledge available on the taxonomy of, as I told you, 16R sequence is the most conserved region. So it is one of the criteria used for the classification of all ever identified phytoplasma so far. And second is RFLP. And on that basis, we have identified 34 authentic groups and different subgroups on the basis of RFLP pattern. This is the classification recent on the recent Burgess manual. Now it is a member of class molecules under Achilioplasmates, because it is having maximum genome sequence similarity with Achilioplasma. That's why you can see it is phylogeny is maximum close similarity with Achilioplasma, 87 to 88.5%. So it is kept under this order and family. These are the just list of groups to, these are the different groups, 16SR, and I have just made a little difference in color just to know that the blue one we have reported from India, and rest all from the rest part of the world. But all they are very, very significant in different crops, and India, so far we have, out of 34, we have already reported 12 groups in India, causing different types of diseases in different crops. This is all the list we have, updated list. Now every day we have been adding, because we have characterizing new phytoplasma. Now we have very good tools available with us. Different parts of the world now people they have started working on phytoplasma, and they have reported new diseases, and on the basis of gene sequences they have been characterizing a particular group or subgroup. for that particular phytoplasma strain. You can see the diversity. You won't find a single crop that is not affected with this phytoplasma. It's starting from fruits, ornamentals, vegetables, cereals, no, oil crops, industrial crops, then pulses, spices, and most of the wheats, they are infected with phytoplasma. So it is really very, very wide diversity of phytoplasma causing diseases to different crops worldwide. Even trees, you can see in India we have only reported, but if you see the world picture, you will find more number of trees are infected with this phytoplasma. Already I told you this is a regular recurrence. For the last several decades, I tell you 4 or 5 decades, we have been facing problem for these important diseases. Already we have discussed in historical discussion that since then it has becoming a problem, and still we are facing a challenge. For sugarcane, because I work more than 25 years in sugarcane, so I have more exposure of sugarcane phytoplasma. Every year we have been evaluating new genotypes for phytoplasma. We don't have any 100% resistant, but we are trying to have some tolerant or moderately tolerant varieties, at least for existence for 2-3 years for commercial cultivation. And again, we have to repeat the process. So that is so cumbersome, but we don't have any other alternative since we don't have any natural resource, resistant resource available. So that is the little management practices we have made adopting, but that is not 100% cure success for management of this disease. Like sesame, already we have discussed, coconut, areca nut, sandal, brinjal, and ornamentals again, herbs, shrubs. Those are having very soft tissues and short duration crop. That are mostly you will find affected with different instances of phytoplasma because of fast multiplication of phytoplasma in them, and their response to that particular crop. And we can see weed species are again very, very important, because it can play a major role in natural propagation. They are very, very potential natural reservoirs and play a very active role. That suppose when your crop is grown, a particular brinjal is harvested. After that time, the same phytoplasma strain is moved to these alternative hosts, complete their life cycle, and again with the help of vector, they can pass on to next season crop. So that's why cycle continues. So weed's role you can see again very, very important for as a natural reservoir. How they transmit in nature, just I am giving a brief introduction first before going to detection procedure. It transmits in nature by vegetative propagation. This means the crops which are vegetatively propagated, chances of phytoplasma occurrence is much more, like sugarcane, grapevine, most of the ornamentals growing by bulb, tubers, or by vegetative cuttings, So dodder is experimental, since it is not, we can't culture it, and it is not very easy to transmit, through insects. So that is the only experimental plant where you can maintain your culture from field to glass house. So that is very, very important. Any plant in the field, if you saw symptom and confirm phytoplasma association, you can transfer it with the help of dodder, that different species of cuscuta, with on catharanthus or any experimental plant, and maintain a culture in the lab, your lab for entire study. So that is very, very useful. Then seed, very, very hardly meager, 4 or 5 instances are available where phytoplasma transmission through seeds are reported. Some tomato, maize, some ornamentals we have reported from India, and coconut only recently is a very important discovery. One of colleague from Kasargod, they reported association of phytoplasma transmission through embryo. So earlier they never believed, how phytoplasma is transmitted through embryo, but they prove experimentally, and now it is very, very dangerous after having this confirmation that it is being, it can transmit through embryo. Then insect vector, I told you, it is the number 1 natural source of transmission, and 3 important vectors, leafhopper, plant hoppers, and xylids. They are known. more than at least 700, 800 species of leafhoppers. Out of these, leafhoppers are number 1 in transmitting, followed by leafhoppers and xylids. Xylids are very, very important in especially fruit crops, horticulture crops, apples, pears, stone fruits, and all other related hard trees. They are very successful in transmitting phytoplasma from infected to healthy one. And then already I discussed about weed. Alternatives are playing not as a weed. Other same, suppose brinjal field is healthy, nearby infected sesame field is there, and same phytoplasma stain may be infected brinjal. That is equal possibility that it can transmit to your nearby brinjal fields and infect the same phytoplasma, and both your sesame and brinjal fields would be infected. So all these are very important criteria which are helping phytoplasma to get transmitted from one source to other. This just to show you different types of grafting. It is being a very popular practice for horticulture crops, ornamentals, and that's why, because there is a phloem to phloem contact, a maximum chances of phytoplasma transmission. You can see grey point seedlings, sugarcane cuttings, and different types of grafting are responsible for phytoplasma transmission. Different types of corn tubers are also known for transmitting phytoplasma. This is a little list of whatever all over the world. We have very limited list available for transmission through seeds, and these are some examples I have displayed here. Winter's aisle seed, tomato, corn seed, alfalfa, pea, and the last one is from India. We have reported here is a celosia ornamental plant. We have reported seeds, but less than 2% transmitted, and coconut root wilt from South India. This is the daughter plant, used as experimental plant for transferring phytoplasma from natural source to your experimental either glass house or poly house. Very commonly, routinely used all over the world, and even it is not only transferring. Suppose in nature, if it is available, infected and nearby, for example, I would like to give example of Iran, where lime witch's broom is very, very important. And lime, what the scientists found, that because they have acreage and acreage of lime cultivation, it is a very commercial product of Iran. And what they found, they are really surprised. Within 6 months, most of the in healthy orchard, all of a sudden most of the lime trees, they were started showing phytoplasma symptoms. So they started working on the real cause behind it, and they found this Caturra natural growing on some of the lime trees. Because there are not much distance between healthy nearby growing orchard trees, they are the cause of transferring phytoplasma from one tree to another. So that way also it is very important. Vectors already I discussed now. They are very, very important source, and lot of research is going on. Now we have, it is very, very little mechanism is known, that it is not possible that one leafhopper is vectored all phytoplasma stains, or all phytoplasma, one phytoplasma stains is vectored by all leafhopper species. Very, very specific. There is a mechanism involved known as antigenic membrane protein microfilament complex is formed, and they cross 2 barriers, salivary gland barrier, and that is another but is very interesting, and now came to know there is a very real molecular involvement of unless until protein complex from phytoplasma and leafhopper is formed, only they then recognize and they admit that particular leafhopper to feed on and ingest that particular phytoplasma strain to reproduce inside insect cell. So it is very, very particular and specific, but again very, very important. And just at this time I would like again to know, because of little climate change for the last 4 or 5 years, we have been realizing the importance of these insects. Little rise in temperature, population is being increased, and from those areas where we never reported any phytoplasma incidents, we started getting report. Sir, we are for the last 4 or 5 years we never had any problem, but now we start all of a sudden. And then I requested them to please visit their field and see whether some leafhopper populations are there. They told yes, sir, because we are leafhopper population are being increased. then I told this is the real cause of phytoplasma incidence in that crop. So that we have to manage that way also. It is very, very important. I told you they can complete their life cycle both in plant and insects. That's again very important, and that is any vector, entire phytoplasma can multiply up to a desired concentration and reach to the salivary gland. That is the storehouse of phytoplasma. And having reached a particular concentration, that is sent to 10 to the power 7 or 4,000 to 6,000 phytoplasma cell per nanogram of insect DNA, then it is capable of infecting, feed on a new plant to initiate the disease. Already we have discussed, we have several vectors we have reported from associated with different diseases all over India. Just a list for your information that this is our, now we have focusing our studies towards epidemiology, characterization, and then host metabolic interaction. Then only we can plan some management strategies. As a natural reservoir, I told you, weeds are playing very important role. More than, in India itself, more than 30 weed species we have reported as a very potential alternate host for at least 7-8 different phytoplasma groups. Some of the list I mentioned here. The red one are very, very common everywhere. Cannabis, for example, Cannabis sativa, Sanadon, Parthenium, Amaranthus, Datura, and Phalanthus. Everywhere where we go, we find this disease, and they are showing symptoms, and they are reported to be associated with phytoplasma. So that is very, very important role of these weed species. Now come to diagnosis. Again, I have added little more picture of symptoms just for the knowledge of the attendees. So at least if you go the field, you can identify and realize, yes, this symptom may be because of phytoplasma. Then you can plan further for further identification. Because symptom is again very, it is not possible that without having any knowledge about symptom, you can collect any sample and directly process for any molecular analysis or genome assisted analysis. You have to have little knowledge of symptom. Either they have not 100% reality idea for direct identification, but yes, it has very, very important, phytoplasma has symptoms. Some of the symptoms of phytoplasma are very, very important, where you can realize, yes, the 90% it may be because phytoplasma. And then you can plan further for this PCR. The first one is symptomatology. These are the common symptoms we observe for different phytoplasma disease combination: yellowing, colosis, little leaf, stunting, phyllody, witch's broom, proliferation of shoots and roots, flat stem, dieback, and different types of malformations, in flowers, fruits, and different parts of the plants. This is the white leaf of sugarcane. You see such an important one, especially in Maharashtra, Tamil Nadu, Uttar Pradesh, Bihar, Haryana, very, very serious disease. And especially, must be most of those who have little idea about sugarcane, we used to grow 2 times, second year also as a ratoon for the plant crop, and we got more incidence in ratoon crop because of this disease. And you can see, incidence of plant 40 to 5%. This is the picture of Western Uttar Pradesh, Shahjahanpur. We have a research, I work over there. So we surveyed regularly fields, and we found every in ratoon crops incidence was much more as compared to the plant crop. You see, there is no 100% loss in ratoon crop, no trilling, no fruiting, and there is no scope of any tonnage of milling cane after this disease infection. This is another phenotype, same phytoplasma strain, 16SR11 group, that is known as Candidatus phytoplasma oryzae. But see, entire different symptoms. Earlier we thought that it may be different phytoplasma strains associated with this phenotypic expression, but hundreds of times we did PCR, we found this is the same. But we found when I visited other countries like Thailand, Vietnam, Bangladesh, most of the time we got only white leaf symptoms. But in India, It may be because of we both have tropic and subtropic environments, and that's why we have different type of phenotypic expression of symptoms. Very recently, 1990, we have reported another disease of phytoplasma. Earlier, sugarcane yellow leaf syndrome earlier, and that is the disease is a mixed infection of a luteovirus, that is sugarcane yellow leaf virus, and this phytoplasma. But since 2019, while surveying, when we collected some samples, we found only association of phytoplasma. Then we identified it as a new disease, sugarcane leaf yellows, that is again caused by 16R first group, that is known as Candidatus phytoplasma asteris. So that's just for your idea, different types of, that is sandal spike, it's very old. You see the healthy tree and the infected one. There is no wood, entire loss of the crop, tonnage of the, because wood is the main economical value part of the tree, and essential oil. Both 100% reduction. There is, you can see in the beginning seedling itself, there is a witch's broom and spike symptoms. So at that time, people confirm using Dean's stain for their identification, but only is such an old disease, but only we confirm phytoplasma association in 1986 only. So this is we are still very lag behind in proper identification of phytoplasma associated with a major disease occurring in India. So that is a challenge. Those who are I discussing one of the colleague from Masood, I told it's a very serious disease, and now we are planning to have more idea about epidemiology, characterization, and then we can plan how we can manage this important disease. Sesame phyllodes, you can see total phyllodes, no flowering, fruiting, and you can see total. And only this work of one of my MSc student, Dr. Sajad is here. He only did entire, we surveyed all over India, collected samples and confirmed association of first and second group as the major cause of sesame phyllodes in India. We also identified the infect vectors and alternative cause for the disease. You can see all over India, it is everywhere it is there, such an important. Ground very little, very recently, we received the samples from Andhra. You can see the little leaf and shoot proliferation from peanut field, very severe. Every year, this one of our student, division student, he was regularly observing this for the last 4 or 5 years. Then he sent some of our samples and we confirm association of phytoplasma. So Thoria phyllodes, you can see the phyllodes, total conversion of floral part into vegetative part. All different types of vegetables you see. You won't find any vegetable where there is no report of phytoplasma, starting from tomato, potato, cabbage, carrot, different types of salad, lettuce, and cabbage, it's all potato, shoot proliferation, cooker beets, chili, culling of leaves. Somebody we are discussing, you observed. Again, earlier most of the time we got association of begomovirus, but now it is very common, because earlier we never attempted phytoplasma, association of phytoplasma. Earlier we, most of the labs we only focus, suppose I am interested in phytoplasma, I used to identify phytoplasma. Somebody is interested in virus, he used to identify virus. But now we have realized in most of the cases, not only one, most of the time we got association of more than 2 virus, more than 2 phytoplasma, and now with the help of next genome sequencing, it is open endless opportunity. You can identify a dozen of virus, dozen of phytoplasma associated with a particular disease. So that is the utilization of this genome assisted diagnosis. So now we have started realizing not only viruses, phytoplasma are equally important in damaging our vegetable crops, and causing very severe losses. Brinjal is number 1, no doubt. You can see there is no scope of any economical value after this infection. Tomato big bud, recently from Andhra and Hyderabad area near around, we have reported this disease, and now we have started getting news of this particular disease from most part of the state where tomato is being grown. although viruses like TOSPO and Begum are very, very important, but now this phytoplasma is also becoming one of the very alarming one in damaging our potential commercial varieties of tomato. Carrot, no. Then all legumes you can see, pigeon pea, cowpea, soya bean, lentil, they are all showing different types of symptoms and infected. Chickpea is again a very recent melody. We can see complex disease again because it is associated with not only one, two different types of viruses and phytoplasma also. And most of North Indian states and southern states, it is showing stun disease, very, very common. Master virus is reported, glaucoma virus is reported, phytoplasma we have reported, and Dr. Reddy is again, he is the main work for his PhD thesis. So we are collecting samples from all chickpea growing areas over India to collect the diversity, to know the diversity of viruses and phytoplasma. So again, a very important disease where phytoplasma is associated. Now come to ornamentals. You see, like vegetables, none of the ornamental will find free of phytoplasma. Some few examples I have here. Jasmine from South Bengaluru, chrysanthemum, you see complete this phyllodes of flowers, and marigold, you see very stunting and specific stunting yellowing, and bougainvillea. So all crops will find infected. This is the experiment Phytocatharanthus. Like dodder, we use an experimental plant for transmission. This is the experimental plant for maintenance of phytoplasma culture, because it's very easily grown and very easy to maintain, and luckily all 34 groups of phytoplasma, they can transfer through dodder or through insect to this plant for maintenance of culture inside lab, inside glass house. See, variety, there is endless, any plant if you name, I will show you the symptoms. So this is the wide diversity and occurrence of phytoplasma. Chrysanthemum, sunflower, flat stem is again very, very common symptoms, and very easy. You will find a very flat stem in some ornamental plant, even vegetables and some other plants, even trees, Zapota, in fruit trees we are getting a flat stem, and all we are found associated with different groups of phytoplasma. Rose, you can see variety of symptoms from IRA field. This is work of my one PhD student. She screened more than 30 varieties and reported phytoplasma in most of the 21. And only earlier they thought it may be physiological disorder or some other, but we confirmed no, because earlier they tested for virus association of fungi, bacteria, and they didn't find association. Then they were very worried, but we confirm, and now we are trying to eradicate and eliminate by using tetracycline and tissue culture by little recovery of these diseases. Various types of malformation you will find in different types of rose varieties. Apple, now come to fruit. Very well, you come international global scenario. If you talk about losses, these are the top 5 important phytoplasma diseases. Millions of euros we are losing because of the disease. All over Europe, American countries, this is very, very important apple proliferation phytoplasma. So far, India, we have very little attempted. Now we are planning, because in Himachal Pradesh, Kashmir, and Uttarakhand, we have apple orchards, and some suspected symptoms we received, but so far no proper studies has been done to characterize the phytoplasma. Dr. Sajad is working on viruses, and in future definitely you will have more knowledge about phytoplasma association in apple from India. Peach, you see, again peach, very, very important. Again, 2 different types of diseases caused by phytoplasma. Peach decline and little leaf are very, very important. They belong to third and X disease, and the first group, that is CAP status group. Zapota, I told you, 4-5 states we surveyed, and phyllodes symptoms, you can see here leaf life structure from Bengaluru and Tripura and others for flat stem, and we got association of phytoplasma. A pineapple, which is broom, strawberry, this fruit and flower, this fruit essence, where chlorophyll pigments are more, and there is no normal development of fruits, and that is all caused by phytoplasma. This symptom you will never find with any bacteria, any fungi or virus disease. So these types of symptoms are very, very common, at least phyllodes, witch's broom, flat stem, and some this type of malformation, no, where more higher pigmentation of chlorophyll. Grape wine, again, no, I talked about the important apple, grape wine. All over Europe, that is the major economical crop for wine production and for table fruit. And that is the only crop very, very susceptible to phytoplasma. And because these orchards, we can't, one orchard grown means that is retained for 30 years. And that's why little phytoplasm multiplies day by day and causes infection, and there's a severe damage because of this phytoplasma in grape wine. Very recently in India, we have identified phytoplasma. From India, we have many states they have started growing. Baramati is one of the main region in India, in Maharashtra, and we have identified phytoplasma stains from reddening of leaves and yellowing. And you must be having knowledge in our Dr. Banmal's group. They have identified first stain viruses and virus from grape wine. Although more than 70 viruses have been reported on grape wine, but India, no earlier, only for the last 2-3 years, only the help of microarray chip, we could able to identify new viruses and virus associated with grape wine. Pomegranate, you see, this is a healthy one, this is an infected one. Guava, yellow leaf, mango malformation. It was again a mango malformation melody is again a very, very, very under confusion. Most of the people, they told some environmental factor, insects are associated, some other abiotic stress. So we are, for the last 4-5 years, we have been trying and trying. And for the regular, we receive samples from Delhi and Tripura and other Punjab area, and we confirmed that it may be other pathogen associated, but one component of phytoplasma is confirmed. So now we have to treat that angle, So it is again a very, very, for the several times for the last 2-3 decades, we are getting instruction from our headquarters that somebody should work because of the importance. Everywhere it is very, very common. And we are losing our fruits because of this malady in most part of mango growing states. But confirm of phytoplasma, only we confirmed recently from India, from 3-4 places. Lime witch's broom, I told you, Iran, very serious. You won't find a single fruit in infected trees. 100% loss is the biggest damage caused by lime witch's broom in Iran. Like we are in sugarcane, sesame and brinjal in India. Rice orange, rice, viruses are again very important. Rice tungro virus. This type are known as rice yellow leaf disease or orange leaf disease. Is symptoms matches with rice tungro. That is caused by 2 different viruses you must be knowing. Rice tungro spherical virus, rice tungro bacilliform virus. And again is caused by a zinc deficiency known as ICAR-IARI disease. They express similar symptoms. So because this we studied from Gorakhpur, nearby Gorakhpur, and we visited one commercial rice field, we got these symptoms. But earlier I have the knowledge, I was at that time I was knowing that these type of symptoms from Vietnam, from Philippines, they have reported to caused by phytoplasma. So I thought to attempt, why not to attempt this, And one very peculiar you can observe, if a virus disease is there or ICAR-IARI disease, some soil deficiency is there, we'll get symptoms all over the field. But here only is in the border only. So then we suspect that maybe because of nearby fields, it is the role of leafhopper or some particular insects that transmitted this phytoplasma from, and then we work and identified phytoplasma. And the vector we reported is nephrotitis, very common rice plant vector. And the disease was reported as 11th group, that is CFP-IARI, that also infects sugarcane. So you also know that also gives you an idea, because in more of nearby fields, the sugarcane was growing. And then we collect samples from sugarcane, and we detected the similar strain. Then we correlate that it may be from sugarcane, this phytoplasma transmit to this virus, rice disease, rice plant. Wheat, no, I told you, these are very recent. In 2012, we have a regional station at Indore, IRI regional station, which is a major station where we grow both durum and this bred wheat varieties. And durum is very, very common. We bred durum at that place, and every second, third year we release commercial varieties which are very popular, no? All over central, not only all over central India, but rest part of India, and also export to several nearby countries. So we observed, no, very peculiar symptoms. And just like the grape vine, more than 54 viruses are reported on wheat. And very serious problem in South America, Australia, Canada, and rest part of America. So we also realized that it may be one of the virus. And little attempts earlier, very sporadic reports are reported earlier of barley yellow drop, wheat streak, which are very important with viruses affecting wheat crops. So we realized, and then on the basis of literature available, we attempted antibody, all the different serology. We requested antibody, we purchased antibody, attempted ELISA and different serological procedures to test whether it is relating, but we didn't. We failed. Then we attempted various PCR published, PCR primers published for different viruses. We attempted all related PCRs for 5-6 viruses, which are very, very important, reported in India and nearby India, but again we failed. Then at that time, there was only one report from China of phytoplasma disease on wheat. So keeping that in mind, we never thought that phytoplasma may infect wheat also. So then we attempted, and to our surprise, we got 100% confirmation of phytoplasma of CAP-IARI and CAP-Asteris. So that is very important one, and at very primitive stage you can see there is no tillering, very unspecific stunting, dropping, no tillering, and besides yellowing and some clotic spots on the leaves of the most of the durum varieties. So it is, suppose it is, we surveyed 100 durum and bred wheat, we got 80 durum genotypes affected with this symptom and only 20. So it is more on, we reported incidence on durum. And again we identified 2 different vector species. Again it is a part of student, PhD student work. Led from Jammu area, we reported earlier from only Yugoslavia, Romania, they have reported this disease. But we got hills area, very, very common, entire hills, they are showing very, because of these reddening symptoms. At a distance one can see. And then we collect such samples, and so it is just for your information. If you see these type of symptoms, you can realize, yes, it may be because of phytoplasma, and accordingly you can plan your further identification work. Now come to coconut. Yeah, I told you very, very important. Lot of investigation, confusion was there earlier. Now it is confirmed that it is only phytoplasma causing this disease. And still 1-2 vector reports are there, but it is not confirmed. Still we have to confirm the vector. But again, there is no remedy to control. Now our station is working for developing some resistant genotypes. Working is going on, but still we have to do a lot for further studies. Erica nut yellow leaf, you can see sometimes there is no top, even after growing 6 months, you will seeing a very prominent leaf following symptoms, and there is no further growth development. Kernels, the real economic, the fruits are affected, nuts are affected, and there is no value in market, So again, it is very, very important. Very recently in Kerala, again we have reported on a spice, a small cardamom. You must be Dr. Jain introduced in his lecture that we have reported 2 new virus diseases from this division, Chirke and Furke disease on large cardamom. But so far, not only from India, not any part of the world, there is no report on phytoplasma. And only last year, 2016-17, one of our colleague working in Kerala Agriculture University, she informed that farmers are reporting some very unique symptoms, very unspecific symptoms like shoot proliferation. And then we received some samples and identified as a phytoplasma from Idukki area of Kerala. So this is just glimpses of symptoms. Just why I added little more, so you have a little more knowledge. If you see those symptoms in the field, you can realize, and you can either, you are most welcome. If you can remember, you can send photographs to me. You can send samples to us for further identification, so that once it is identified, then because it's not possible for everything we can do from here, but you can do help in identification, then you can complete this part of study like epidemiology, losses, other biological studies in your area just to complete the study, so that we could have planned a very, very relevant and potential way of management of these new diseases. So now come to diagnosis. Second, after having knowledge of symptoms, after now staining. In earlier days where no electron microscopy and serology and molecular biology was there, only two major staining techniques were used. One is Dean's stain, very, very common. Even the sample you are handling here, you can cut with razor, very, here, and stain with Dean's stain, and we can observe the only these stains are very specific to phloem tissues. No, they can stain with blue color, and along with healthy, you can compare and identify. Yes, this is maybe infected, but 100% confirmation only we can confirm after molecular this genome assisted diagnosis. Like Dean's two stains are there. No, it's very simple preparation. Methylene blue and azure and maltose. Just mix in 100 ml of distilled water. Simply, those who are having no facility, at least you can begin identification of suspect of, suppose you have sent samples to me. By the time you can, at least you can get an idea whether yes, phytoplasma they are not. With help of simple microscope, 40x magnification, very clearly you can observe difference between healthy and infected tissues by staining this Dean's stain. Likewise, there is fluorescence, a nucleic acid is specific stain, where you can only phytoplasma cells with phloem, this phytoplasma bodies inside phloem tissues is fluoresced with this stain, and you can clearly distinguish with the healthy and infected one. This is the leaf, and this is the leaf petiole. So these are still they are very authentic techniques using all over the world where facilities are not available, or just to confirm on a very primary basis. Electron microscope, where electron microscopy is there, you can 100% you can see the presence of phytoplasma pleomorphic bodies in TEM transmission electron microscopy. But again, it is not possible, it is not available everywhere. And not only for this, it is not easy. Just a virus, for virus sample, you take a drop of juice and put on grid and see the virus particle. Here you have to cut the very ultra thin section with the help of ultramicrotome. Large other these protocols are there to fix in glutaraldehyde, and then only you can see in this electron microscope. But again, very, very authentic, and 100% confirmation of phytoplasma detection by using electron microscopy. Again, a very recent discovery. This is all new for us. Still, we are having knowledge that it is not culturable. But as we say for today, it is culturable. Little beginning starts, but we have to confirm. There is no commercial, this product is available in the market. It is under patent condition, but Italy group of scientists, they have got success in culturing this group of microRNA. You can see how it grows non-culture media. Very complex media. I have given just a brief composition, but it is not 100% composition. The major component they have added to culture these bacteria. So it is a challenge for us, and I request all of you also, very easy. You can go through, because mycoplasma are culturable. Uroplasma, many other mycoplasmas infecting medicals and animals, they are culturable. So now we can take knowledge of those media and we can utilize, if anybody having facilities for culture, you can start your career for phyto. Is again very, very important, challenging job. If one could develop a media for phytoplasma, you will be well known, you will be recognized worldwide. Again, very challenging, very simple. You can attempt daily, but it is a regular job. You have to keep different media, different concentration, whatever knowledge available. So again, it is a very, very good work initiative to work on phytoplasma. Serological identification, viruses, very, very significant. But phytoplasma, no. Because why? We don't have a code protein, very specific protein, like there is no cell wall, only antigenic membrane protein. Some successful attempts were there. They isolated the EMP protein, then purify the protein and develop either polyclonal antibody or monoclonal antibody. But for Iran, I told you, they got very good success. They developed this ELISA-based test, then dot blot immunoassay, and they developed lateral flow kits also for field. Field, it is very easy. If you can take a lateral flow and just drop 2-3 drops of infected, you will get a reaction within 5 minutes. So that type of work we wanted here. We should initiate in India for the 6 major diseases we are having, on different crops. Now come genome basis. Most of you are well aware for the for 4 or 5 days we have been doing DNA extraction. It is little special in the case of phytoplasma. Why? Because we don't have 100%, even after having very good symptoms in the field, you can't assume that titer will be very high. So you have to adapt, and lot of secondary metabolites, polyphenols and other contaminants of plants, they disturb the quality extraction of DNA, phytoplasma DNA from the plant. So several protocols are available, and we have also modifying, every lab is modifying, it's standardizing its own protocol for isolation of DNA, extraction of DNA for a particular plant. Then followed by PCR, you are doing we need a primer. Already universal primers are there, but it is not the end, We are dealing with different strains of phytoplasma. So always there is a scope to develop new set of primers for a better and suitable amplification of a phytoplasma strain with less concentration. So again, there is always a scope for designing new, and luckily we have a full genome sequence available with 7 strains of phytoplasma. So you can, and free softwares for primer designing are available. So you can utilize and develop as your own phytoplasma strain specific primer. Suppose you are working on a particular crop, you can have sequence of that phytoplasma, design your own primer for a very suitable and very easy amplification with even less titer of DNA. So it is going nested PCR only. That is not only used for phytoplasma. We can use for viruses also, other diseases, pathogens also. But here its relevance is more because titer is less. So first time in first round, most of the time we never get any amplification. So we designed another internal set of primers, giving more specific and sensitive to amplify more of the more of the strand of that particular specific gene, for example 16SR, and chances are more. So you will get very clear amplification in nested PCR. That is routinely used in phytoplasma. That you will do tomorrow morning, followed by gel electrophoresis, very normal we do. After then amplification kits are available. We elute and purify the from the gel, and then clone the product. Cloning is important to have the full length of amplified product, because generally is a lot of error, handle error, and we can't get a full length. So, but again it is cost effective, you have to pay more. But only for describing a new pathogen, new sequence, you have to have clone. Otherwise you won't publish your paper in a good journal. For having an idea is okay, you can directly sequence and have a yes, it related to this one. But for publication, for authentic identification, you have to have cloned your product for authentic sequence length, And then sequencing, a lot of softwares for this editing and editing the sequence for submission. And then once you have sequence, then you can compare with different tools available. That is again very important for analyzing data for identification of any type of sequences, bioinformatic tools. And luckily we have lectured by one of a colleague where we will be exposed to different bioinformatic tools that can be utilized for sequence analysis of data. 5 minutes. So this is just again how DNA, again you have for especially phytoplasma, because it resides in phloem tissues. So you have to take care which tissue you are taking for DNA extraction. That is very small things were very important. For example, you know that it is only resides in phloem. So if you select the midrib of the leaf or young growing shoots with rich phloem tissues, chances are better for better DNA extraction. And likewise, if you are from insect you are collecting, generally we use the entire insect body, but if you are able to separate the salivary gland, which is the storehouse where maximum phytoplasma titer is there, chances are better. And very easy. Once it's a very small technique. With any entomology expert, you can learn how to isolate the salivary gland from the insect. 3-4 insects you can collect, isolate the salivary gland, and then process for DNA. The 100% your DNA, this quality will be improved. And for older trees, we can take bark, where phloem tissues are much. I told you nested PCR. Generally, this is universal PCR. We are using P1, P7, what we are utilizing today. And tomorrow we will use 2 different sets of nested PCR. It is not only one standard. Varieties of first round and second round universal PCRs are available. You have to standardize with your phytoplasma state, with your expertise, with your facility available with you. So a lot of is already there, and every day we are modifying depending upon we are facing some problem. We are adding as per other publication. So we in our lab we found if we add a little peg, we got in tissues where a lot of latex and polyphenols are there, we got very good amplification. This is a list of primers we are used. This is not the only list. The 100 of lists are available, published from different labs. But we have standardized from India. We have designed these 2 10 years back, and it is working very well. 90% of the cases with 3F, 3R, and IF7, we are getting very good amplification. So you can try PCR amplification with these primers, and definitely you will get better results. These are the cycles we are using today. Just more or less. Only we have to little analytic temperature we have to think of. You have to standardize your own PCR cycle protocol by simply, you can't change 5 degree, 6 degree. By simply varying a degree or 2, you can better amplification, and you can standardize the protocol for your lab detection. This mixture already you are using. What are the components? The very important one is magnesium chloride, DNTPs, and this enzyme, a reverse primer, forward primer, and template DNA. So this component you have to take, all should be in better quality, so you can get better expectation of results. Nested PCR already we discussed. These are the results how you do. You get the first round and second round. Very clear bands, there is no confusion. And then you can identify, you can isolate, get it purified, eluted, and get it sequenced for further comparison studies. I told these are the 7 full genome sequence available for phytoplasma all over the world. The last 2 one is from India. Very recently, 2 months back, only submitted on NCBI. And I think there are 4-5 full genome sequence in pipeline. So within 2-3 months, the list is, every day list is increasing. And that's make you a better possibility of exploring the whole genome, different genes involved in the phytoplasma genome for further study. Besides this, although it is a very conserved region, 16SR region, after having full genome sequence available, we have several scientists targeted different multi-locus genes on phytoplasma genome for very finer classification of phytoplasma. And they found that there is 20, 30 genes are there but these genes are most routinely used for very clear-cut taxonomic differentiation and resolution of phytoplasma strains by using ribosomal protein gene, SEC-A, SEC-Y, TUF, GROL, immunogenic membrane protein, and garase A, garase B. So daily we have been trying, but these first RP gene, SEC-Y, SEC-A is a very, very better. Why? But because they are having only 60 to 80% similarity with 16SR, two groups of phytoplasma. Suppose there are 16SR1 and 2 group. With comparing 16SR sequence amplification, there is 98%, 80 to 98% similarity. But by same, if you amplify with RP gene, we'll get sequence with two different groups, only up to 60 to 80%. So it's provide you more opportunity for a wider differentiation with the same under same group. Suppose with the help of 16SR first group, we identified 16SR first group, one group with 16SR. But when apply the same amplification with RP gene, we'll find RP groups under 16SR first group. RP group 1, RP group 2 under 16SR first A group. So that gives you ample opportunity for finer differentiation and classification of phytoplasmas. I told you these are the, just I have mentioned what is the property. You can see even in one subgroup they are able to differentiate. So that is the utility of these specific ribosomal protein. These especially there are two of the ribosomal proteins are routinely used, RPL22 and RPS3, and these are some protocols. And you can see it has only 69 to 79% similarity of between two different groups. However, 16SR first, they are having more than 88 to 98%. Just like this is a candidate for SEC-A gene. Again, you can see 69 to 7 and 84% between two. So again, ample opportunity to use this specific primer for better differentiation, finer classification of the same group and subgroup of phytoplasmas. Like SEC-Y gene, this garase, very recently for the first time from here we have on the basis of full genome sequence of oyster, this onion yellows phytoplasma, we designed this primer and utilized and very successfully identification of several phytoplasma strains, garase A and garase B. They are housekeeping genes of phytoplasma genome. TUF gene. So after so these are the genes are very, very applicable in finer differentiation of phytoplasma. Again, after you have amplified, then you have to clone, you have to purify, you have to elute, and then you have to analyze the sequence. For different softwares are there, is a free softwares are available online. Once you have submit your sequence, then you can compare and then you can identify your isolate, whether it is a new one, how much similarity with the old one, how much difference from the other reported previous systems. So many softwares, and that is again important. By the using this bioinformatics software, you can analyze your result in a more better way. Similar results by using different softwares, you can analyze your data more potentially, more significantly for publication and identification of and characterization of particular strains. So bioinformatics tools are again very, very important. Utilization of bioinformatics tools for any sequence analysis, whether it is pathogen, phytoplasma, virus, fungi, or bacteria. Then RFLP, and I told you two groups, relevance of RFLP. They have again, what we do, different 17 endonuclease restriction endonucleases are used for characterization of all the 34 identified phytoplasma groups. And by using this, so far more than 200 subgroups have been reported. So it is more, if you compare with 16SR classification that are more popularly utilized. And out of these 17, I have listed here, the 3, HIN-FL, MSC and RS1 is the most popular one. This is the phylogeny again, you can sequence your phylogeny with using mega softwares and analyze your data by clustering. It gives an idea about your relationship for phytoplasma with earlier reported one. Two different types of RLP. It is virtual and real. Virtual means there is a computer programs are there. Simply we have to load your sequence and you will get your results within 10 minutes. Developed by USDA scientists known as I5 classifier tool. No? It is developed by Zhao et al. In 1990s. No? Very, once we have sequence, simply we have to put in the software. You will get the similar coefficient value, which group it belongs, which subgroup it belongs. It is very easy. No? But it is just a tentative idea. It's not 100%, but it will give a tentative primitive idea of association of your strain. But this is a real RFLP. After PCR, in the same mixture, where you are doing, you have to add restriction enzymes and run. You will get the similar results what we have seen in this I5 classifier tool, and you will get the same. So this is more authentic. You can compare for a better publication in a good journal. You have to do RFLP, real RFLP to prove your results for final classification of any phytoplasma strain. See, I told you the 3 major enzymes. No, only these 3 major enzymes, they are capable of differentiating all the subgroups of the 14 groups of phytoplasma. So out of 13, suppose it is not possible because they are costly. So at least for beginning, you can analyze RFLP with these 3 at least to give an idea of differentiation of strains. And if budget available, you can apply all the different enzymes for comparison and analysis. After this, it's just an idea like viruses, real time we also use in. It is not routinely used for phytoplasma, but it is again well established for several phytoplasma strains where we can identify for each cycle with the help of different this color signal, you can quantify the phytoplasma in each run of the cycle. However, in conventional PCR, only at the last you will get. So that is the difference. With the help of quantify, we don't know by simply showing the symptoms and what is the whether titer is high or less. But with the help of this, you can quantify the phytoplasma in any infected plant sample. That is the utility. Multiplex PCR is also used. Yesterday also you heard and discussed. Here also we can use the same protocol. By using the same PCR mixture, you can, instead of 1 primer, you can add 2 set of primers. You can add virus primer also. Simply we have to standardize the annealing temperature little, this cycle, different component of the cycle, and you can standardize your protocol for identification of a virus and phytoplasma, mixture of viruses, mixture of phytoplasma in one go. So that is the utility, just like yesterday Dr. Jain explained. Loop mediated, just like viruses also, it is isothermal recent development where you don't need any thermal cycle, thermocycler. Simply at a room 60 to 65 degrees centigrade with 3 and 4 set of primers, you can apply very fast, very sensitive. This, and it is filled. You can take this to field and develop. Very small machine is there. With only you need incubator, and you can develop the result, and you can check your results by coming to lab. And there is also another fluorescent dye you can use, and simply by visuality, either by magnesium pyrophosphate turbidity is there, or cybergene, you can see a different color for phytoplasma presence, just like other pathogens, fungi, bacteria, and viruses. Recombinase polymerase, again more recent developed one, within only 15-20 minutes with the 2 set of primers, you can have the reaction. That is the utility of these isothermal reaction without any much laboratory facility. So at least you could try. These are the recent genome assisted techniques. No need of even DNA extraction. Simply by plant extracts, even you can initiate the reaction and got the confirmation. So these are the utility. They are very fast developing, very sensitive, very authentic, 10 to 100 times sensitive than PCR. So you can realize the significance of these techniques. That is all about. Just in a brief, how, since it is a challenge, we don't have any proper management strategy to plant management for any of the phytoplasma experiments so far. So whatever little success with us, resistant variety is definitely one. We have very little success. Only, for example, I explained in sugarcane, every year we evaluate variety, develop varieties for mortality resistance and resistance, and we can, for other also, brinjal, we have wild relatives for sesame and brinjal, we have been used, crossed, and we got a very little success. But really we have to move forward to have more approach, molecular approach for management, Vector control is very important, but as you know, chemical, everybody is banning chemical pesticides, and because of environmental concern, we have to shift to biological control approach. And epiphytes are coming up very good way. Other control by other insects, their spider species have been identified, which are eating eggs of leafhoppers, plant hoppers. So like that, agronomic practices, little, if you know incidence of phytoplasma vectors, you can little late or sow your crop little early or little late, just to avoid the first and second influence of leafhopper population and manage your crop. Like control of weed crop, I told you, weed are playing very important role. If you know the symptoms on weed, immediately you have to eradicate rogue up and save your crops. Only tetracycline is effective. And in ornamental crop, we got very good success by applying 60 or 80 ppm at fortnightly we sprayed in field, and we got very good removal of symptoms. And because short duration crop, and so we can have very good this flower for commercial value, and farmers can retain their crop for commercial market. Tissue culture, for little eradication purpose we are using because it is not growing as fast as in younger parts of the plant. So apical meristem we can using for successful management of in some of the vegetatively propagated crop. Hot water treatment is very effective. For sugarcane, recommended 52 degrees centigrade for 2 hours because of little hard rind of the sugarcane crop. But most of the ornamental and grapevine, where the softer tissues, we recommend 50 degrees for 2 hours. And it's very, very effective for management of phytoplasma. Then quarantine, again, a very real concern. We all know, we are educated people. Still, we don't follow quarantine regulation. Without, we are taking any sample from one place to another place. We are eating fruits and taking to another place, and that's why disposing is spreading new types of diseases in newer areas. So that is again, at least we are aware people, educated people, we should guide and educate other society just to avoid. This is very important concern. If you go to any foreign countries, then you can realize the importance of quarantine, how strict it is. You can't move, you can't take a single piece of seed or any piece of the plant or living tissues without permission. So that is again very relevant. I told you this is like ICTV. In phytoplasma, there is the International Phytoplasma. This is the website. You can visit regularly, and you can all the information on activities of phytoplasma worldwide. So those who are interested in phytoplasma, you can visit this website to know the various types of conferences, publication, projects, and people working on different groups of phytoplasma. So very important. I'm also a member of this group. And as Dr. Pan told, we have recently edited 3 books. It is the updated information of all different types of identification, epidemiology, management, host metabolic interaction, and genome information development on phytoplasma. So again is a recent book. In ISI, but it will take little more time. You are most welcome to submit your publications related to phytoplasma, spiroplasma, candidatus vector to this journal. Thank you very much. Sorry for little